Hello, Biology 1 student. This lesson will focus on Competency 5, Heredity. I have included the performance level descriptors for informational purposes only. Here you will find your assessment verbs and scientific terms associated with Competency 5. Before we get started with the lesson, I would like for you to download and complete the full square vocabulary sheet for assessment verbs and scientific terms that are unfamiliar to you. This lesson is designed to review the Competency 5 Biology 1 content you learned in the classroom. Let's start with the central dogma of molecular biology. By that, we mean the flow of DNA to proteins. DNA is transcribed into RNA through a process called transcription. RNA makes proteins through a process called translation. So there you will see the flow of information. DNA, RNA to proteins. That is called the central dogma of molecular biology. Here you will find scientists associated with the race to discover the structure of DNA. Pauling, Chargaff, Wilkins, Franklin, Watson, and Crick. Let's review the structure of DNA. DNA basic shape is a double helix, or it looks like a twisted letter. As you can see from the diagram below. DNA is a nucleic acid made up of nucleotides. Each nucleotide is composed of three parts. You have a sugar that's called deoxyribose, you have a phosphate group, and you have 4 nitrogen-containing bases. Adenine, which is usually represented by the letter A, thymine, T, guanine, C, and cytosine, C. The picture at the bottom shows you a typical nucleotide. 3-part unit, phosphate, sugar, and base. DNA has 4 kind of bases, which we've already discussed. Hydrogen bonds form between adenine and thymine, as well as between cytosine and guanine. At the bottom, you will find a diagram of DNA nucleotides. Let's continue with the structure of DNA. Chargaff analyzed the amount of adenine, guanine, thymine, and cytosine in the DNA of various species. He found that the amount of guanine nearly equals the amount of cytosine, and the amount of adenine nearly equals the amount of thymine within a species. Adenine and guanine are purine bases. Cytosine and thymine are pyrimidine bases. And you will see the base pairing in the diagrams at the bottom. Let's look at base pair rules. Purines only pair with pyrimidines. 3 hydrogen bonds are required to bond guanine and cytosine. Examine the diagram below. 2 hydrogen bonds are required to bond adenine and thymine, as you can see in the diagram. So let's remember that G bonds to C and A bonds to T. Let's complete the practice activity. Fill in the blank to create complementary strands of DNA. Remember, A bonds to T and G bonds to C. So let's take a moment to complete this activity. And here are your answers. Again, noting that A bonds to T and G bonds to C. This slide shows you the percentage of bases in 4 organisms. Take a moment to review the information. What do these data suggest to you? It should show you that the percentages of adenine and thymine bases are almost equal in any sample of DNA. The same thing is true for the percentages of cytosine and guanine. DNA replicates itself. DNA replication is the process that ensures that each resulting cell has the same complete set of DNA molecules. During replication, the DNA molecule separates into two strands and then produces two new complementary strands following the rules of base pairing. An example is listed below. Genes contain coded DNA structure that tells cells how to build proteins. Remember, DNA is in the nucleus and proteins are made on the ribosomes. So somehow the genetic code in DNA must get from the nucleus to the ribosomes. This happens because of a process called transcription. Transcription is the process by which a molecule of DNA is copied into a complementary strand of RNA. RNA then uses the base sequence copied from DNA to direct the production of proteins. Here you will find a chart comparing RNA to DNA. The sugar in RNA is called ribose. The sugar in DNA is called deoxyribose. RNA is single-stranded. DNA is double-stranded. Uracil has replaced thymine in RNA, so therefore the bases in RNA will be A, G, C, and U. The bases in DNA, as we stated earlier, would be A, G, C, and T. There are three types of RNA. Messenger RNA carries the genetic message of DNA from the nucleus to other parts of the cell. Ribosomal RNA forms parts of ribosomes. And transfer RNA transfers each amino acid to the ribosome as it is specified by the coded message in messenger RNA. The first step in decoding genetic messages is to transcribe a nucleotide base sequence from DNA to RNA. This transcribed information contains a code for making protein. Remember, RNA contains 4 different bases: adenine, cytosine, guanine, and uracil. These bases form a language or genetic code with just 4 letters. Each 3-letter word in messenger RNA is known as a codon. A codon consists of 3 consecutive bases that specify a single amino acid to be added to the protein chain. Anticodons are the 3 unpaired bases of transfer RNA. These bases are complementary to messenger RNA. Let's practice reading codons. Suppose you wanted to determine which amino acid is encoded by the CAU codon. Find the first base C in the left column. Next, find the second base A in the top row. Find the box where these two intersect. Find the third base, U, in the right column. Find the box where all 3 intersect. CAU codes for histidine, abbreviated HIS. Here are some genetic terms for review. Remember, homozygous genotype is a gene combination involving 2 dominant or recessive genes. In other words, the genes are the same. Heterozygous is a gene combination of 1 dominant and 1 recessive gene, also called a hybrid. Dominant is the stronger of the 2 genes expressed in the hybrid, represented by a capital letter. Recessive is a gene that shows up less often in a cross, represented by a lowercase letter. Punnett squares. Punnett squares use mathematical probability to help predict the genotype and phenotype combinations in genetic crosses. And here's an example of the Punnett square at the bottom. Let's examine how to make a Punnett square. Punnett squares allow geneticists to predict possible genotypes and phenotypes of offspring. In this example, both parents are heterozygous for yellow P allele. First step, make the grid. Place the alleles of the gametes of one parent along the top of a grid and those of the other parent along the left-hand side, as you can see in the diagram. Step 2, fill in the grid. Combine the parent alleles inside the boxes. The letters show the genotypes of the offspring. The genotype ratio is 1:1, meaning 1 big Y, capital Y, 2 capital Y, little y, 1 little y, little y. The genotype ratio is 1:1, meaning 1 capital Y, 2 capital Y, lowercase y, 1 lowercase y, lowercase y. Step 3, fill in the offspring. Use the law of dominance to determine the phenotypes and phenotype ratio of the offspring. As you can see, has been labeled for you in the diagram. The phenotype ratio is 3:1, meaning 3 yellow peas to 1 green pea. There is an additional example of a Punnett square at the bottom of the page. Here are additional examples of monohybrid crosses. Take a moment to study these diagrams. There are some inheritance patterns that would not follow Mendel's laws. The first one we're going to review is incomplete dominance. In incomplete dominance, neither allele is not completely dominant over the other. The heterozygous phenotype is an intermediate phenotype between the two homozygous phenotypes. Some textbooks describe this as the blending. In this particular case, you will see red and white flowers producing pink flowers. In codominance, an organism that has both alleles of a gene displays both phenotype at the same time. An example of this is a cross between a black cat and a tan cat, and it results in a new color, in this particular case, a cat that is called Tabby. Another example of that would be the roan color you see at the top of the page. Many genes have multiple alleles, including the human gene for blood type. And here you will find the genotypes and phenotypes for blood type. Also, you will see an example of how to complete a Punnett square. In the example shown, you will find a mother with type AB blood mating with a father with type B blood, producing offspring with type AB as well as type B blood. Sex-linked genes are located on the X chromosomes. Traits determined by sex-linked genes are called sex-linked trait. Color blindness is an example of a sex-linked trait. A carrier is a person that has the trait on only one chromosome and does not express the trait. Carriers of sex-linked traits are always women. An example of a colorblind female carrier is X big C, X little c. Let's practice for a moment. Eye color is a sex-linked trait in fruit flies. Complete the chart below. Here are your answers. XRR is a red-eyed female. X big R, X little r is a red-eyed female, that indicating that the female is the carrier. A white-eyed female will have the genotype X little r, X little r. X big RY is a red-eyed male, and the genotype for A white-eyed male would be X little r Y. A karyotype is a visual representation of an individual's chromosome. A karyotype shows the complete diploid set of chromosomes grouped together in pairs which are arranged in order of decreasing size. Here you will find a karyotype of a normal male. We know that it's a male because it has X and Y chromosomes. Also, you will find a karyotype of an individual with Down syndrome. This is also called trisomy 21. If you notice, there's an extra chromosome at the 21st pair. Let's take a moment to look at this karyotype. Can you determine whether or not it's a male or a female? It is a male because of the X and Y chromosome. A pedigree is a diagram that traces the inheritance of a particular trait through several generations. A pedigree uses symbols to illustrate the inheritance of a trait. Here you will find some of your common pedigree symbols. At this time, I would like for you to check available downloads to complete Competency 5 lesson.